But we then wanted to be able to manipulate these cells. So it was interesting that they are activated, whatever that means, but we wanted to know if actually modulating these cells made a difference to the transition to chronic CRPS, specifically to the pain phenotypes that we're able to detect. So we use this trick of glial cell turnover compared to peripheral myeloid turnover. And what I mean by that is that, as I mentioned, myeloid cells include both peripheral macrophages and microglia. But importantly, microglia are very slow to turn over. And in normal conditions, they're not replenished by peripheral cells. So they only can replenish from internal stores. And so what we did was we used this CX3CR1 CRE mouse. And first just confirming that we actually see a change in turnover using a TD tomato mouse, so that when we give tamoxifen, we turn on the tomato, and that initially we'll see microglia that are both labeled green because of the CX3CR1 receptor, but also red because of TDT expression. Same with the macrophages. But as the macrophages turn over very fast and the microglia turn over very slowly, if at all, we would then lose expression of the TD tomato and in the macrophages and only see the dual color microglia. So we wanted to first confirm that that was actually the case, and that it was tamoxifen dependent. So you can see here, this is again spinal cord dorsal horn, and without tamoxifen, we don't see any TD tomato. With tamoxifen, we see beautiful expression of TD tomato in these CX3CR1 cells. And then what we did is we looked at the turnover. So we looked at 3 days versus 24 days post-tamoxifen, and with TD tomato by flow cytometry. And you can see here in the top is the CNS, and you see this persistent TD tomato expression out to 24 days after tamoxifen. Whereas in the macrophages from the spleen, there's TD tomato expression initially, but after 24 days, these cells have turned over and they no longer express the TD tomato. So we were pretty confident that using these CX3CR1 CRE mice, we could differentiate the effects of microglia from those of macrophages. So we took further advantage of that by generating these mice, again, with the same CX3CR1 driver, but this time using a diphtheria toxin receptor that would be expressed under the CRE gene. And so we basically developed these mice that when the tamoxifen activated CRE enters the nucleus, the stop codons are moved and you get expression of the diphtheria toxin receptor. Again, it's going to be in both microglia and macrophages, but then once macrophages turn over, we get expression of that receptor only in microglia, so that when we dose with diphtheria toxin, we're only going to deplete microglia. So again, it allows us to really separate out the effects of the peripheral from the central cells. So again, the first thing we wanted to do was just to make sure that this was actually the case. So at baseline, again, you could see here the spinal cord dorsal horn and a beautiful microglial cell with many different branches. But at day 1 after diphtheria toxin, there's just almost completely devoid of microglial cells here. And those that remain look different. They're smaller and have less branching. And then over time after depletion, they do repopulate, which is really important. And I'll tell you why in a minute, but we're really interested. They repopulate from these central stores where the few remaining microglia actually manage to repopulate the entire CNS. And we're looking here just at the number of microglia over time. We also performed shoal analysis just to confirm that they do increase in complexity over time. At baseline, there being many branches, and we can look at the number of intersections from the radial distance from the soma. And we see here that, again, the day 1 microglia are much less complex compared to those at day 14. And when we look at an area under the curve, we can see that. Interestingly, though, at day 14, these cells still remain slightly different from their baseline. So they're not exactly the same microglia that were there initially. We also just looked to make sure that the depletion alone didn't affect any behaviors, and it didn't. But that's not so exciting. But what was exciting was we developed these somewhat complex behavioral schemes to be able to deplete microglia at different time points after cast removal or during this CRPS progression. So in this case, we depleted microglia at the time of the injury. Then we also looked at what would happen if we depleted microglia at this key, what we think of this 3-week time point, this acute to chronic transition. And then we also decided to deplete microglia at this sort of later phase, so really into the chronic phase at 7 weeks. And what we found was really interesting. So when we depleted the microglia right at the time of casting, we see here, so again, this is mechanical threshold along the y-axis and time after fracture on the x-axis. So all of these mice developed sensitivity, but if we just look at allodynia as the readout, both the males and the females that received the microglia depletion at the time of injury had a reversal, although it wasn't complete, but there was an improved trajectory that was significantly different from the non-depleted mice. When we did it at the chronic phase, so at this sort of late phase, 7 weeks, we saw that these mice all developed very significant sensitivity, but after the depletion occurred, they improved, but then they resensitized. And so it seemed almost like by 7 weeks, the microglia had made their contribution and had, triggered subsequent downstream modulators to then take over. The most impressive data we found, though, was when we depleted at this 3-week acute, what we were thinking of as the acute to chronic phase transition. And what we saw here was that these mice all started off sensitive, but both in males and females after fracture, in the depleted groups, we saw a complete reversal of trajectory, which was very exciting to us because it was probably the most striking data that we've seen in terms of an actual change in a behavior after modulation of really anything. So we've seen some changes where it goes from maybe 0.1 to 0.2, but here we actually saw that it went from a very low mechanical threshold up to about 1.5 grams. And then the other interesting thing was that we saw improvement in hot plate latency, both in males and females, improvement in weight bearing in males and females. So now they're feeling maybe more comfortable. They can walk on that injured limb. And very interestingly, we actually saw an improvement in these peripheral signs related to the CRPS injury. So we saw an improvement in the swelling and an improvement in the temperature differential between the injured and the non-injured limb, which was quite surprising because it suggests that central modulation of neuroinflammation can actually affect peripheral outcomes, which we were quite excited about. So right now what we're doing is looking at transcriptomics of these repopulated microglia. So as I mentioned, at about 14 days, the microglia are here, they're back. They look a little bit different than they did at baseline. And we're trying to understand what that difference is to really understand if those microglia are carrying some sort of signature that could then be reproduced as a therapeutic. And so we're taking those cells, as I said, that are repopulated. We're doing FACS sorting using a sort of complex but very highly specific microglia isolation protocol. And we're deep into the bioinformatics now, trying to figure out with RNA sequencing what these populations look like. And so hopefully the next time I talk to you, we'll have more information on that. But just so that I can match a little bit of what Ted was talking about earlier, the other thing that we're doing is also looking at human spinal cord microglia. And so we have a collaboration with Donor Network West, which is a local transplant organization here in Northern California. And we actually go to the operating room ourselves and harvest this tissue when there is a donor available, for which we're very grateful. And we are taking these lumbar spinal cord tissue and performing single nuclei isolation, and then looking to isolate spinal cord dorsal horn microglia and perform single nuclei sequencing, mostly because we want to understand if what we're seeing in the mice is actually reproduced in the humans, because ultimately, if we come up with the most amazing mouse microglia target and humans don't express it, then it's perhaps not that interesting. So we're trying to use this as a way to validate our targets as we move forward with this project. And so, Stephanie, I don't know how much time I have left, so I maybe just check with you before I.
Yeah, we are going until 5 Eastern, so, and we want to leave some time for questions. Maybe we'll just leave a few minutes at the end. So I think you have another 5 or 10 minutes if you need it.
So I'm just gonna, thank you. I lost track of time because I was so into Ted's talk earlier, so I didn't know what time I started. So I'll just tell you a little bit about sort of I guess it's a famous story in the microglia world, which is the story of TLR4. So I think at this point we're convinced that microglia contribute to the acute to chronic pain transition in CRPS, and again, specific to the mechanism of this one condition, but maybe it can expand to others. And it certainly has similar effects from what we see in both males and females. But that has been controversial because not everyone has seen an effect of microglia in both males and females. So we were really interested to understand that maybe perhaps there are different ways that they'd be activated. These are complex cells, they're not all or none, and they have many different receptors that are expressed. So TLR4 became this sort of hot topic about 20 years ago now when I was actually in Joyce DeLeo's lab, that it might be a trigger for microglial activation. It's expressed by all myeloid lineage cells, and actually it's also expressed by neurons and astrocytes. It's the canonical receptor for LPS, which is found on gram-negative bacteria. And there were several studies that showed that actually if you gave LPS, you could produce allodynia or sensitivity in males, but not females. And a paper that Joyce published when I was in the lab back in 2005 showed that mice that were deficient or dominant negative for the TLR4 receptor had less sensitivity. And this is kind of an opposite graph because it's number of withdrawals to a stimulus, so less pain being a lower number. And so they showed that TLR4 knockouts had less pain. So it seemed like a potential target. And then a nice paper that followed several years later by Robert Sorge when he was at Jeff Mogul showed again that LPS injection was effective at decreasing threshold in males only and not females, and that this was in a TLR4 dependent fashion. So we were interested in understanding this a little bit better because nobody had really looked at TLR4 specifically in microglia. The mouse models that were used were done with dominant negatives. These were germline changes. They weren't specific to microglia. So the first thing we did was just try to reproduce the prior data. And so what we did was we gave a TLR4 antagonist called LPSRS, and we dosed it intrathecally. And we just followed the hours after injection and tested mechanical sensitivity. And you can see here that the males had an improvement in mechanical sensitivity. And it's very, very close to Robert Sorge's data. So I was like, this like never happens. But it was great because we were really able to reproduce what he saw, which was that in males, you can see here in the male control mouse, there's a really nice reversal of that mechanical allodynia using this compound. But we wanted to take advantage of this mouse that we had already been using, as I mentioned in the depletion study, the CX3CR1 Cree mouse, and we mated it to a TLR floxed mouse to get a conditional knockout that would be both time and cell specific. So one interesting thing that came from this work is when we isolated whole CNS, we found that the tamoxifen injection resulted in a significant decrease in TLR4 expression, but there was still some TLR4 expression in the whole CNS. However, when we FACS isolated microglia, there was a massive drop down to essentially undetectable levels, suggesting that other cells besides microglia do express TLR4 in the CNS. And this has been known and has been shown, but the antibodies for TLR4 were never that good. So it made us feel, pretty happy when we found this result. So we were confident that we could get microglia specific TLR4 knockout. And again, using our trick of time, we can make sure that it was microglia only and not myeloid lineage cells. So we did our same sort of procedure where we gave tamoxifen early, we waited for the peripheral cells to repopulate, and then we did our fracture in the same CRPS model. And interestingly, in the mice that had the early TLR4 knockout, the males had a complete reversal of trajectory in their mechanical responses and their allodynia. And while the females had a significant response or improvement, it was not as striking as in the males. When we did this at that later time point, so we did it so that the TLR4 knockout was sort of coming online at 3 weeks at that acute to chronic transition, we saw that in both males and females, there was a reversal, but again, not nearly as impressive as what we saw with the early knockout.